Anxiety is a word, a concept that everybody is familiar with. It's a natural response in anticipation of something that might be dangerous.
When anxiety, do you think that becomes a problem?
Anxiety becomes pathological when it's really affecting your daily life and has negative consequences. Patients with anxiety disorder, a certain brain region will be overactivated when you present them aversive stimulation. Two regions really stand up as really strongly overactivated, and these are the amygdala, and another region is the insular cortex. So our work is exploring a pathway that has never been studied so far. Depending on where the dopamine is released, it can have different function based on the downstream neurons and where these neurons will project again.
You're listening to the Neurochemistry Podcast, presented to you by the International Society for Neurochemistry. Thanks for tuning in. Let's get started.
So, hello everyone. Thanks for being here with us today. The Neurochemistry Podcast, where neurochemistry is for everyone. I'm Will Sanchez, and today our really special guest is Dr. Anna Beyeler. She's a full professor at the Institut National pour la Santé et la Recherche Médicale INSERM at the University of Bordeaux in France. So thanks, Anna, for being here with us today.
Thank you. Thank you very much for having me. Just for the general public to have an idea of what INSERM is, it's actually the French Institute of Health.
Good. That's really helpful for us.
Very good accent, but maybe everybody doesn't know.
Sure. Well, we gonna start talking a little bit about anxiety and all around the research that involves anxiety. But maybe we can start this conversation telling the people what anxiety is, and more than that, is that why is a biological reason or evolutionary reason, if you will, to anxiety to exist, right?
Well, that's actually a very good question, because anxiety is a word, a concept that everybody is familiar with, is using, but it actually has a reason why it's there, and everybody's not always agreeing on what it is. So it's a natural response in anticipation of something that might be dangerous, right? So it's different from fear, where fear would be a reaction to a danger that is present. So it's very important in an evolutionary perspective, because, if you are only able to be afraid and avoid danger when they're there, your likelihood of survival is much lower. Whether if you are able to anticipate some dangers, or for example, you could see like the footprint of a predator, or just the smell of a predator, and say, oh, maybe I shouldn't go there. And then so this anticipation of danger will allow you to avoid this. And so mainly it would be to protect your life.
So if anxiety increases, like fear in stress, anxiety, that increases your chances to survive in something like life-threatening. When anxiety, do you think that becomes a problem when it's not something that natural?
Well, that's a good point. So you have a spectrum of anxiety. So every animal, including humans, will have a different level of anxiety. And, that's your innate level of anxiety, or in psychiatry, it's called a trait anxiety. And this is very variable. So I guess evolutionary, that will be preparing for different environment and situations. So increasing the likelihood of animals to survive, because indeed, if you're too anxious and then you never go out of your house, whatever animal you are, then you cannot get food and you cannot reproduce. So that would also affect your survival. And in an evolutionary perspective, that is also not good. So in humans, if you think of that, we have different standards. We are not at survival anymore, but we are more at well-being expectations. And anxiety becomes pathological when it's really affecting your daily life and has negative consequences on your daily life. So it could be that it's impacting your social life, so you cannot go out anymore because you're afraid of talking to people. This is different of the fear of crowds, right? It's really like social interaction, fear of social interaction. So this is one type of anxiety disorder, the social anxiety disorder. So you have different types of anxiety disorder. And you have another type which is very common, which is generalized anxiety disorder. And one of the symptoms for that one would be that people don't want to go out for work because they're feeling too anxious about it, of every complicated or bad thing that could happen there. So this is also something that could be used as a symptom to diagnose anxiety disorder.
So between all the things that we know and we don't know about anxiety and we want to know, what do you think that are the key brain areas that are mostly, assigned to the anxiety behaviors and anxiety circuitry in the brain? Because people probably want to know a little bit about it.
Well, yeah, that's very interesting, and it's really the heart of my work and why I go to work every morning. So there are studies that have been done in humans using imaging, fMRI, and it was shown that in patients with anxiety disorder, a certain brain region will be overactivated when you present them aversive stimulation compared to the general population. And in these, there are a network of region, but two region really stand up as really strongly overactivated, and these are the amygdala, which has been well studied for its implication in fear and also anxiety. And another region is the insular cortex, which is also very interesting because it's the region that relays interoceptive signal from your body. And very few, or let's say much fewer studies have been focusing on the insular cortex. So my lab is dedicated to studying the contribution of the insular cortex in anxiety at a very fine level, so at cellular and circuit level.
So you touched that part on the human studies, but we know that we do also, preclinical studies. So do you see some similarities between, animal models as well as in humans?
Yeah, so actually, the first publications of my lab have shown that the insular cortex is also active when animals are in anxiogenic situations. So we have specific mazes for little mice, and when we record the activity within this brain region, the insular cortex, we also call it insula. So the neural activity in the insula is increased when the mice are exploring an open area where they would be vulnerable to predators, for example.
What happened in the insula, it's neurochemical changes that produce an activation of different cells. Are we talking about neurons or some different cells?
So here we're talking specifically of neurons, and even more than that, we're talking about excitatory neurons. So neurons that will release glutamate to activate downstream region in the brain. And we also dissected the connection from the insula to the amygdala, because these two regions are strongly connected in both directions. And we assess the output from the insula to the amygdala. And this circuit is also increased. The activity within these neurons is also increased when they explore anxiogenic spaces. And if you change the activity of these neurons, you will increase anxiety.
I couldn't avoid to notice that in your lab page, you have published the insular cortex, anxiety, and dopamine. So it was kind of not surprising to me, but I want you to talk a little bit because, dopamine, like in general public and the scientific community, we usually saw that dopamine is related to reward, motivation, maybe movement, regarding anything that is motivated related behaviors, as well as in memory that are linked to something rewarding. So could you tell us a little bit more about dopamine, how it is involved in anxiety in the insula?
Yeah, so that's a very exciting paper. That is actually going to be the point of my talk tomorrow at the meeting. So we have a preprint, so it's not fully published yet. I mean, it's in peer review process currently. So historically, dopamine, as you were mentioning, has not been studied from a negative valence point of view, and it was really reward or motor function. But some in the early 70s, some clinical observation have shown that patients with Parkinson's disease or patients that receive treatment for Parkinson's disease, which are modulating dopamine transmission, had changes in their levels of anxiety. So that was a first observation. And then in parallel, some really old school pharmacological manipulation of the receptors of dopamine, which are of two types, the D1 and D2. The D1 like family would be like more activating the neurons that express them, and the D2 would be more inhibiting them. And these studies have shown that you can also change the level of anxiety by specifically targeting one or the D1 or the D2 receptor. And what is very interesting is that depending on which brain region you manipulate this receptor, you will have different effects. And so it had never been done in the insular cortex. And we have shown that if you activate the D1 receptor, this will increase anxiety in mice.
So let's imagine that I'm a mouse, right? I'm entering some specific context that it's anxiogenic for me. That's going to create anxiety. So that's going to produce an increase in dopamine that going to activate those neurons that you told, the neurons that express that receptor, D1 receptor. So it's correlated with that.
Yes,
right?
That is correct. But it's even more than that, because if we block this receptor in the anterior insular cortex, then you will decrease anxiety.
So it's safe to say that depending on where we are seeing dopamine release or increasing dopamine, we're going to see different outcomes in the behavior, right?
And this is like work that has been published in the early 2000s by Stéphane Lamel. And he beautifully showed that in the VTA, which is the brain region where most dopamine of the brain is produced. I mean, most, like with the substantia nigra, but more of the mesolimbic dopamine system. And there you have different pathway out to the brain. So, for example, to the nucleus accumbens or to the prefrontal cortex. And these two stream will have different function. And so our work is kind of in line with what has been previously shown, but exploring a pathway that has never been studied so far. So yes, it's again validating that depending on where the dopamine is released, it can have different function based on the downstream neurons and where these neurons will project again.
So you said about at the beginning that mostly historically has been studied anxiety and negative kind of states, emotional states in the amygdala. Does the amygdala and the insular cortex have any connections between them?
Yeah, they have actually very strong connection. So so far we did not map whether these neurons from the insula that project to the amygdala have more D1 or more D2 receptor. So this we don't know yet. But what we know is that neurons that are excitatory or inhibitory within the insula will have different amounts of the receptors, the D1 or D2 receptors.
So probably people have to think about, well, what does it mean in terms of translational knowledge in treatment for anxiety? What do you think that is more relevant in terms of going to the clinic with those results, if possible?
I guess the main contribution of this work is to highlight again that it's not as simple, and you cannot put the brain in a bath of a specific pharmacological agent because it's so diverse. So we need to think of different ways to change the activity of specific circuits, because indeed, if you give the antagonist for the D1 receptor, it might act in the insular cortex and then decrease anxiety, but it will also act on many other circuits and have side effects. So this is really the limit of the current pharmacotherapies for all psychiatric disorders, actually. So the idea would be to find ways to specifically manipulate circuits. So you have invasive approaches, which could be chemogenetics, but they are indeed quite invasive, and also in terms of how much they would cost, that would be difficult to put in place. And there are also ethical issues because they use DNA vectors. Another approach, which is still very early on, would be to try to use neurofeedback. So have a way to monitor the activity of a circuit and then have a visual or any sensory feedback to the patient that could train to decrease the activity of the circuit. that could be. But these are very long-term strategies, right? It's not going to help patients tomorrow. I think how it can help patients to actually already today is to understand that there's really a biological nature to their troubles, and that we are working on identifying what is working differently in the brain of patients suffering from anxiety disorders, and hopefully in the next decade we can find ways to restore that.
Medicinal chemistry have like evolved huge in these years, so maybe, I don't know what you think about it, but the people can design drugs that are more specific for some brain area sometimes. So like bitopic ligands or ligands that can act on some receptors in the brain. Maybe that is also a key, I don't know what you think about it, but it's a key part of the research to give to the bridges, or to build bridges from the preclinical to the clinical part, so they can build new pharmacological treatments or know a little bit better about behavioral treatments as well.
No, that's a good point. And indeed, there is a huge amount of work we could do at the molecular level. But for the dopamine system itself, directly for the study, like the D1 receptor is the same in all the different regions. So it would be really difficult to target it specifically in the insula. So there are also strategies where you could think of a release of a molecule specifically in the insula, like triggered by ultrasound, for example, or something like that. I'm not an expert of this field, but I know there is research in that direction, and this could be very promising. And indeed, or research could directly inform what we could test in clinical trials.
Well, during this conversation and all the results that you touched a little bit about, and all the fundamental terms and everything is surrounding anxiety, we might have some clues. But can you say or summarize something about why it's so important to invest in science, especially for our case and for the purpose of this podcast, for neurochemistry or neurochemical projects? What do you think about it?
Well, I think that the first level is really understanding about nature that is surrounding us. And sometimes, you search for something and you find something else, and that can completely revolutionize the way we are thinking of a system, or we are going to treat a disease that we were initially not studying. So I think exploratory research is fundamental, because, this is how, in the end, we're going to treat the patients and save lives. And then, of course, you want to also have studies dedicated to certain pathologies, and these are also very important. And the ratio of turning a fundamental research paper to a treatment it's very low. But if you look at the larger scale and long term, you have progression, and the evolution of our understanding has completely metamorphosed the way we are treating patients with psychiatric disorder today compared to 50 years ago. So these are very important. And here I'm speaking to all the people that suffer from a psychiatric disorder or have a close one that suffers from it. And it is fundamental to understand what is biologically different to be able to treat it, and only fundamental research can do that.
Well, when we do science, we have several ways or several opportunities to help at the end people or the environment. That's probably one of our goals in our scientific careers. But we're probably going to have people that suffer from anxiety. So do you want to send them any message? Like,
maybe what I would say is that every morning when I go to work, it's really thinking of them and hoping we can. And I'm not the only one. There's many people around the world like working on anxiety in preclinical and in clinical settings. And we are investing a lot of resources to understand, and we have made a lot of progress. We're not at easy and fast therapy, but there is very promising ways, like the use of psychedelics now are really changing the way we're thinking, because these drugs can, when they're really well set up, they can change and induce plasticity in the brain and have long-term effects. And this is very different from what has been done in the past, because so far the treatments are chronic treatments, and very often if you stop the treatment, then the symptoms come back. And this new family of chemicals that act more on the serotonin system seem to have long-term effects. So that could be actually a new way, and maybe an opening towards curing patients. But
Well, that last comment open a kind of a question maybe for some people that are hearing this. What is plasticity, brain plasticity? So what does it mean? Because we can see that even in the pop culture, that you have songs about that, about dopamine, about serotonin and stuff. But what is plasticity in brain?
so plasticity would be a change in the brain that lasts. So this is in opposition to something that is elastic, that would come back to the original position afterwards. Plasticity, you will have a change and it will remain in this state. So this is actually what we are hoping for when we treat patients. So when I was talking about neurofeedback, the idea is to teach the brain or the patient to decrease the activity or increase the activity of a selective circuit. And with the psychedelics, you could use another angle and use a pharmacological approach to do that, and induce changes that will be beneficial for the patient and will be long term.
So why psychedelics right now are like just being touched again, since I don't know how many years before we had that kind of idea and research behind that, and like as went through, and right now we're coming back with psychedelics. Why is so important?
Well, there was indeed a first big wave of research on psychedelics, because the beneficial effects on anxiety and depression especially had been identified, but there were also drugs that were used recreationally, and then the U.S. government put a ban on the use of these drugs and cut all research funding on these drugs. So yeah, for about 50 years, like nothing happened. And now, we are seeing that we didn't find another magical drug that would treat these disorders. And so, science has pushed the evidence, and mainly clinical assays have shown that it has antidepressive effects and it has anxiolytic effects long term. So it's very important to explore. And in the lab, we have another project which is still not out there, but where we try to dissect the mechanism of action of psychedelics, of certain psychedelics, because it's a whole family in the insular cortex.
That was something that I want to point it, is that depending on your diagnosis and depending on the psychedelic, we still need to do a little bit more research to understand that. It's not that someone that is listening, I have anxiety, so I'm going to use whatever psychedelics is in the market, right?
Yes, it's a whole family of molecules, and you also have side effects that depending on the dose you're using, you can have hallucinations, which are also could be very problematic. So you need to have this in a controlled setting. Taking the drug is like in a hospital, like a little bit like ketamine. It's not that you can take the ketamine at your home, right? It's really controlled posology. I don't know if you can say this word in English.
And it seems that it's also like a promising treatment, not only for anxiety, but also for depression, for, some traumas, still post-traumatic disorders.
Indeed.
That's something that is promising, but still we need like a lot
of we are really, there's like multiple countries. I think it's 5 countries now in the world where you can use psychedelics to treat patients. So in the US, it's not in the entire US, it's only certain states that have it. But it's really growing, and there are international networks to study that. And I see that in the next year, we're going to know much more, and maybe this is another way that we can, and maybe some psychedelics, because so far they're being studied on their impact on serotonin, but it's not impossible that some of them, I mean, it's known that some of them also act on dopamine with a much lower affinity, but still, and maybe the combination of these two action on serotonin and dopamine could be the cause or the reason for their beneficial effects.
Got it. So it's also to monitor also the how rewarding it can be, because it can be have like abuse liabilities. So then, well, we still need to understand a little bit more about that. But it's nice that we have something that is promising for everyone that is suffering from anxiety for the purpose of this episode. And I'm gonna ask you something, because we maybe gonna have young scientists that probably are following you and say your name. Oh, it's Hannah. So well, let's see what she wants to say about this. But also, I want to take the opportunity to ask you, what do you think for a new or a young scientist, that like there's a huge generation of scientists that are coming here, that gonna pursue the academic career, right? So what is one or two advices you want to tell them about that?
That's a very important question. So first, I would like to say that one thing I really love about this meeting, ISN, is that it's really open to the young scientists, and it's very supportive to them. And I think this is critical for like building the next generation of neuroscientists. And if I could say an advice, I mean, I got asked a few times this question, and I think it all comes down to being positive is really one of the most important key, and especially in those times. I'm thinking of the people doing research in the US. It might be difficult, but it's a job that you can do in most countries of the world, so you can travel and discover other culture, other way of doing the science, and so don't get shut down by the problem like short term. And it's like multiple times in my career I was like, okay, that's done. I'm stopping now. I'm quitting. It's too hard. And don't let these obstacles, like really learn from them. Like, what brought you to this situation? Why did you fail somehow? And continue and try to see what you can. What can make you stronger in this experience. So be positive, I think is really key. There's always something good in the situation you are going through.
Well, before we move on to the next couple of questions, which are the final, I forgot to ask you, gender matters? Is gender a factor in anxiety, independent of the kind of anxiety that you're talking about?
Yeah, so in humans, it's very strong. So you have about twice more women than men that will be diagnosed with anxiety disorders. So it matters a lot. Like, it's very similar for depression. So it's a huge factor. It's still unclear how much of this difference is due to biological changes or societal changes. So there's also kind of a social construct that will support women go and seeking for help, while men would be expected to keep their feelings for themselves and not seek for help, which could be very detrimental. But it is that. So it's in humans, it's quite unclear what is generating and to what extent this change. And in preclinical models, I mean, of course, they're not humans; it's animal models. But in mice, if you look at the general, if you take a strain that is more or less anxious, it will be roughly the same in male and female. It's just that the male will be more variable, and we think it's because of the hierarchical status of the males. And we thought that female could be more variable depending on their ovarian cycles, but actually no, they're more reliable in terms of really anxiety-related behaviors. Then if you impose a stress and try to generate a model for a pathological level of anxiety, then female might be more vulnerable, but it also depends on the model you're using.
Got it. But you talk about sometimes men try to repress their feelings because of this society, but also the other way around. Sometimes women have a lot of pressure because of the role that the society expects in some points for them. So it's something that we still need to collect some data to analyze, to have evidence, to understand sex differences or gender differences better.
Yeah, absolutely. Like it's not so the thing is that in human, it's not only about biology. There's other factors, and culture and societal constructs are really powerful. And it highlights again the power of your mind and what your brain can change on your behavior. And indeed, like, more research is needed at both level, preclinical and clinical.
Okay, so one of our goals with this podcast is also to understand how neurochemistry works around the world. So how do you see neuroscience, neurochemistry in France regarding investment, amount of work that is done there, and also diversity? How do you see those factors?
Well, I think in Europe, and I will talk mainly about France, which is where I started my lab, there is a lot of support. Like, there are young researchers that are recruited. The system is a bit different, and it's evolving right now, but it had been historically more like a PI would have a lab, and then, to become PI, you should first be a scientist in the lab of this PI, and then you become the head of the lab when they retire. So it tempers somehow the creativity of the young researchers. So now they're balancing that with some startup packages or tenure track position, as in the US. So I think this brings a new breath, and it's growing. So we're actually recruiting at our department currently in Bordeaux. So it's a computational neuroscience position. So if you're interested. In terms of diversity, well, it's also based on the pool of applicants. And I'm very lucky in the institute where we are. We are half men and women PI. But if I look around in France, it's still heavily men team leaders. And so there is still work to be done on that. And I think mainly it's on the pool of applicants. You need to find a way to promote women and people from underrepresented countries to apply and to get the education to the level of application. So there is progress and still work to be done.
We want that trend to still growing up, right?
Yeah, exactly.
Sure. Outside the lab, what Anna likes to do? Hobbies, spend the time. What do you like to do?
Well, I like to do some sports. Like, I used to climb, but recently I had a baby, so I don't climb that much anymore. Yeah, I'm actually here with my partner and my baby.
Oh, that's good. That's also nice. So you can bring your family to this. Maybe I'm with my wife too. So it's really cool that you have.
Yeah, but right now they're wondering in New York, they're not at the conference.
Oh, of course.
I think she's 9 months old. It's a bit early to understand neurochemistry, but soon she will.
I mean, the river is a better view.
Yeah. No, but so now this is family is taking a lot of my time, but I love nature, hiking, exploring. Hiking is something I really appreciate, to be connected to nature. And one of my biggest concern right now is the state of nature and how humans are degrading the environment. And I also see progress in that field, but there's still a lot more work to do. So I'm trying to control my or environmental linked anxiety.
Yeah, we all suffer at some point of anxiety. So it's something we can deal with that. So, well, that was just the last question. Thanks, Anna, again, for accepting this invitation. We're really happy when you accept, because this is a topic that everyone needs to hear about it. So thank you very much.
Thank you so much for having me.
Yeah, so with that, we're going to close this recording. Thank you very much for listening to us, and see you in the next episode.
Thank you for listening. Don't forget to follow and subscribe for more neurochemistry talks. Till next time.